All right, here we go for today. Sorry. And today we're going to be looking at non-Mendelian inheritance. And so these are how most genetic traits are inherited, and they don't quite fit Mendel's laws. So Mendel's laws, remember, were came up with from pea plants and constituted the law of independent assortment and segregation. And things could be nicely predicted by really simple Punnett squares, and it was pretty straightforward. But the idea is that pea plants are really genetically simple, and that made them great to be able to work with and uncover the basics of genetics. But most traits in pea plants are only controlled by a single gene with only two alleles that have dominant alleles and recessive alleles. The actual relationship between genotypes and phenotypes for many other genetic traits is rarely that simple. And so we need to kind of extend the basic ideas of Mendelian genetics to more complex examples. So there's going to be a lot that's similar to what we just talked about Mendelian genetics, and we'll be able to model most with some modified Punnett squares, but they aren't completely explained by Mendel's observations. So one example is called incomplete dominance, where the heterozygous individual, okay, so the individual that has one dominant allele and one recessive allele, has an intermediate or blended phenotype between the two homozygous genotypes. So in these flowers, these carnations, the red flowers have two dominant alleles, and the white flowers have two recessive alleles, just like we would expect in Mendelian genetics. But the difference comes in the heterozygous individual. That is pink. It's a mix, a blend between the two homozygotes. So we would expect normally this to be red, this homozygous dominant organism to be red. We'd expect this homozygous recessive organism to be red, but in Mendelian genetics, we'd expect the heterozygous individual to be red too. In incomplete dominance, we have three different phenotypes for three different genotypes. And so that's a big difference that we can use to kind of determine whether something is incompletely dominant or not. You might see your book use two different letters to represent the alleles. I would suggest not doing it this way, because this can get really confusing, primarily because a lot of the times when you see a different letter, it means there's a different trait. And so I would stick to kind of the way that it's written out on the left right here. So in this cross, what you saw was two homozygous flowers that would produce all heterozygous offspring. So all of their offspring would be pink. And then if those were self-pollinated, you would see a 1 to 2 to 1 ratio, 25% red, 25% white, 50% pink. And so this could tell you that something interesting is going on, just like looking at ratios and Punnett squares for Mendelian traits. A similar phenomenon, and this is kind of easily confused, so we're going to make sure to explain this really well, is called codominance, where instead of the heterozygous individual being a mix between the two homozygous genotypes, the heterozygote has both of their alleles expressed completely. They're not blended at all. So again, the difference is in the heterozygote, but in this case, both alleles are being expressed completely. So let's take a look at what we would have as an example here. The human blood group, so ABO blood groups, is a great example of codominance because there are three alleles. That's not necessary to have codominance, but blood type is also another good example of what we call multiple alleles, when more than two alleles or two versions of a trait are possible. So there's the IA, IB, and I allele. And the IA and IB alleles are called codominant because if both are present, both are expressed. If someone with AB blood has both A type and B type glycoprotein antigens on the surface of the red blood cells, both are produced. And then the I allele is recessive to both A and B. So if we make a table, if you had either of these two genotypes, you would have type A blood. Either of these two genotypes, you'd have type B blood. This codominant genotype would give you AB, and having both recessive I alleles would give you O. Okay? And so when we go through some practice questions together on Zoom, we're going to do quite a few that have to do with blood type because blood type has been a really common type of question on the AP exam as of recently. Another type of non-Mendelian inheritance is called pleiotropy. And most genes are pleiotropic. That means that one gene affects more than one phenotypic characteristic. One gene affects more than one trait. So dwarfism or achondroplasia and gigantism or acromegaly are both examples of pleiotropy at work, where one gene that underlies those conditions affects many different traits that collectively result in what is observed in achondroplasia or in acromegaly. So for example, man, when I was a kid, I really liked the old days of WWE. At that point, it was WWF wrestling. And André the Giant was awesome, right? You might remember him as the actor in The Princess Bride, really huge film. But he had acromegaly, and so the constellation of different traits, including his stature, all of those traits resulted from a specific gene that caused acromegaly. Similarly, achondroplasia is a constellation of different symptoms that are resultant from a specific gene, a pleiotropic gene. And achondroplasia is inherited dominantly. So, as an example, if one individual that had achondroplasia and one individual that didn't have offspring, they would have a 50% chance of having offspring that had achondroplasia. On the other hand, two individuals that are heterozygous for it have a 50% chance of having offspring that have achondroplasia, 25% chance of having kind of some normal offspring. And when an individual inherits both dominant alleles for achondroplasia, it's lethal. That I don't know, that zygote can never mature uh, past being in the womb. They're never born. Okay, so this is if a trait that one might go and see, like we'll talk about tomorrow, genetic counselors for to see their risk factor for passing it along and things like that. Epistasis, so as an example, like in mice or in the next slide, like in Labrador retrievers, is when one gene completely masks another. Not talking about alleles, like that's dominance when one allele masks the other, but this is whole genes. So for example, coat color in mice are two separate genes, completely independent of one another. But if we will see, the C gene can completely mask the color or B gene. So if a mouse would have the dominant copy of the C gene, they would be pigmented. But if they have both recessive copies, they would have no pigment. And then assuming that they're pigmented, if they have a dominant B allele, they're black, and if they have two recessive B alleles, they're brown. So therefore, we could have three possible genotypes. We would expect a dihybrid cross in Mendelian inheritance would give us a 9:3:3:1 phenotypic ratio. Here, it gives us a 9:3:4 ratio. So just looking at the difference of those ratios can help us see that this isn't inherited in a Mendelian way at all. However, if you understand how these two genes collectively function, we can as well make a dihybrid Punnett square, and we can figure out the genotypes and the phenotypes of the offspring. Epistasis in like Labrador retrievers is another example. Kind of the same thing. They have a pigment allele and then they have a concentration allele, and it gives rise to these four really distinctive colorations that we see really commonly in Labrador retrievers. Another way that genes may be inherited or traits may be explained by non-Mendelian inheritance is called polygenic traits. So some phenotypes are determined by the additive or their combined effects of two or more genes on a single characteristic. So things like human traits like skin color, height, weight, intelligence, behavior, all of these things are the result of a mix of multiple, sometimes dozens of different genes all together, and it produces a whole spectrum or continuum of different phenotypes as far as skin shades and heights and weights and things like that. These are really, really hard to predict and to explain because you need to detail the exact functionality of each of the different genes, and then more importantly, how the phenotype is changed by interactions between any two of the alleles that are present that go towards that trait. And so exploring the inheritance of polygenic traits are really, really, really difficult. Right. And then kind of one of the last things we want to mention is sex-linked traits. So these have to do with inheritance of genes that are found on the sex chromosomes, the X and the Y chromosomes, as opposed to what we call the autosomes, or the other 22 pair of chromosomes that aren't the sex chromosomes. It was first kind of discovered at Columbia University by T.H. Morgan and when he was breeding Drosophila or fruit flies, and noticed that some characteristics in fruit flies were what we call sex or X-linked. So here's a karyotype looking at all of the different types of the chromosomes. All of these, 1 through 22, those set of homologous chromosomes, those are called the autosomes. And then these, the last pair, the 23rd pair, is called the sex chromosomes. So sex-linked traits have to do with genes that are on these chromosomes. So in the discovery by T.H. Morgan at Columbia University of sex-linked traits in fruit flies, he ended up breeding a homozygous red-eyed female with a white-eyed male. And in the offspring, he saw that all of the F1 offspring had red eyes. And so when he took one male and one female from that generation and bred them, he noticed something specifically different in the second generation. 100% of only the females had red eyes, but 50% of only the males had red eyes and the other 50% had white. So these traits, the inheritance seemed to depend on whether the flies were male or female. So the complement of sex chromosomes one had seemed to make a difference about one's phenotype. So if we represent this by a Punnett square, we would say that, okay, well, what just normal Mendelian genetics, maybe red is dominant, white is recessive, and so when you cross the red homozygous red female fruit fly and homozygous white male fruit fly, yeah, that's why there was 100% of the offspring that were red, is because red was dominant and white was recessive, because we got all heterozygotes. But why then, when we bred two fruit flies from the F1 generation, we would expect to have a ratio of 3 red to 1 white. But that's not what happened. This is what we would expect. That's not what happened. What we ended up with is 100% female that were red and 50% female or male that were red. Doesn't really add up. So in humans and other animals, there are 2 sex chromosomes, the X and the Y. 2 X chromosomes typically develop into what we biologically assign at birth is female, and 1 X and a Y typically are assigned to male. In organisms with 2 X's, there's a genetic redundancy, like autosomal chromosomes. You have 2 copies of each chromosome, or 2 copies of each allele because there's 2 X chromosomes. Just like you have 2 chromosome number 1s and 2 chromosome number 2s. However, in an X and a Y chromosome, there's no redundancy. The alleles that are on the X chromosome are not on the Y chromosome, and the alleles that are on the Y chromosome are not on the X chromosome. So you only get a maximum of 1 copy of each allele. So when we're looking at sex-linked traits, we are going to do Punnett squares that include us writing out the X's and the Y's for the male and the female parent. Okay, this is just showing you, okay, when a male and a female have children, there's a 50% chance of being female and a 50% chance of being male. So how does this new information kind of help explain the fly experiment? What if we write in the chromosomes, the X and the Y? What if we do that? So this red-eyed female has 2 X chromosomes, and we're saying the red eye allele is on the X chromosome. So we put 2 superscript R's next to each X, representing that this is an X chromosome with the red allele, X chromosome with a red allele, and since the white fruit fly, the male fruit fly, only has 1 X chromosome, we put a white allele on that X chromosome and nothing on the Y allele or chromosome, because the Y chromosome, since it's not an X chromosome, doesn't have the eye color allele on it. So when we do a Punnett square, we still see that we get 100% of F1 offspring having red eyes. All the females are heterozygous, and every male only has, since they only have 1 copy of the X chromosome, only will receive the dominant R allele. Then when we cross 2 fruit flies from the F1 generation, this is where we get, okay, 100% of the female flies will receive this X chromosome from the father that has a dominant R allele, so all of them will be red eyes. But only 50% of the male fruit flies will be red-eyed, because there's only a 1 in 2 chance of receiving the dominant R X chromosome from the female. And that's why we get those ratios that we saw in Morgan's experiments. So if we talk about what genes are on the sex chromosomes, on the Y chromosome there's very few genes, not very many, except for those that in what's called the sex determining region. And it, has been called in the literature the master regulator for maleness. I think that that's a pretty outdated term. What it means is that in that region of the Y chromosome is where the genes for the production of many of male hormones are present. So testosterone and androgens and things like that. On the X chromosome, however, there are other genes and other traits beyond like sex determination and hormones. So, for example, like traits for color blindness and hemophilia or Duchenne's muscular dystrophy, all of those things are sex-linked traits. And if you notice, that's why sex-linked traits, if there's a disease or condition that's sex-linked, males are more likely to have that trait than females, since they will be affected with only one allele. So that's why I think to yourself, if you know someone that is colorblind, they are most likely male, because it is much easier. It's much easy. I can't talk. It's much more. I'm sorry. Males much more easily inherit X-linked conditions rather than females, since they only require one allele to be present to have that condition. So, for example, here's a picture of the human X chromosome and all of the different kind of traits that are found on it. So all of these things are much more easily passed on to female or males than females.